Cool. All right. Good morning, everyone. Thanks for being here on time early on a Friday. Hope everyone's last week of classes, first week of classes is wrapping up good. Yeah, it's always a little bit weird the first week because you're kind of doing stuff in classes, but you're kind of not. Anyway, hope it's going well for everybody. Today we are diving into our first topic of the semester, which is embryology, and we'll spend probably about half a lecture to 3/4 of a lecture on embryology. It's very, very brief overview, but it's really important to understand how the structures of the adult body, it's very important to understand the origins of the structures of the adult body. And to do that, we need to look back to the body before it's an adult. So with that in mind, today's agenda looks like this. The first thing, this is a little out of order. The first thing we're going to do is answer your questions based on the introduction survey. Some of you had some nice questions that are kind of relevant to all of us. So I just want to go over those so we have a clear understanding. Then we're going to test your knowledge of the syllabus with a class Kahoot. As I mentioned earlier, the winner of this Kahoot will achieve a great esteem amongst your peers, and that's important. Then I'm going to lecture on embryology. I have a short activity built into that lecture as well, and then I'll wrap things up. And we'll start our class tradition of getting a Venn diagram at the end of class every Friday. And so today's the first Friday of the semester, so you get your first Friday Venn diagram. So let's answer your questions. So these are just a few that I picked out from the intro surveys that I think all of you might be interested in. So one of you wrote, I learned really well with mnemonics. So if you have any, can you share them? The answer to this is a big yes. Throughout the semester, I'll try to share as many mnemonic devices to remember some of these things as much as possible. And this will probably come up in lab more than lecture, but it's in both. Your textbook also has some mnemonic devices. For example, we want to remember how many vertebrae are in the cervical, thoracic, and lumbar region. And so there's a way to kind of remember how many vertebrae are in each of those regions, 7, 12, and 5. And so we'll deal with lots of those things throughout the semester. Next question, and a few of you wrote something similar, is that you're an athlete and you're going to miss some classes due to games and things like that. The answer to this question is to communicate early and often, and we'll work together to make things up. All of the lectures will be recorded and posted. It's important. It helps me help you if you let me know in advance, but then also remind me. So if you have your schedule now and you know that on October 17th, you're going to be out of town for a game, let me know now. We can kind of think about that ahead of time, but then please do remind me, the week before or the class before so I can make arrangements for you. In general, you'll be able to make up all the work from the lecture just fine with the videos. I can help you do that. You can stop into open hours. Where it gets a little more challenging is if you miss lab times on Tuesdays, but even so, we can work and make that up, and you can work with your lab partners and do that. It's never been an issue before. So just communicate, let me know, and we'll figure it out. Sound good? Cool. And the last one here is how much time should we spend studying? Of course, I can't give you a specific answer to this. I can't say, 37 minutes every day is going to be ideal because it's going to vary amongst all of you and everyone has their own style. But my recommendation, and actually I'm channeling this through Rory Gilmore, who's probably the best student around. That reference is probably a little outdated at this point. But I would recommend studying a little bit, but very often. So for example, 30 minutes every day or something like that. I recommend against waiting until before an exam and saying, okay, I'm going to spend all of Saturday afternoon studying and try to study for 4 hours. It's just not a way to retain information. And especially in this class where it's a lot of memorization, taking things a little chunks at a time, but often is going to be much more beneficial than trying to cram a whole bunch at once. And I'll work in small assignments throughout the semester to kind of help you do that. But really the best thing is just to keep up on the readings and to do a little bit at a time. And it also helps you not feel overwhelmed. If you set literally you can set a timer and say, okay, I'm going to study for 25 minutes today. And you sit down and you open your book and you study and look over your notes. Your timer goes off after 25 minutes, boom, done. It gives you a sense of finality and completion, and then allows you to go around, the rest of your day doing stuff. So I think it's a good strategy. I highly recommend. If anyone wants to get into that in more detail or anything, you can drop by my open hours on Monday. I'd be happy to kind of work out a study schedule or, whatever that can. Anyone else have any tips on studying for a class like this that's worked for you? Yeah, so good. Flashcards. Okay, tell us about how do you do flashcards?
Usually I group them together like on categories, like doing like bones, cavities, that sort of thing, and color them. Very visual kind of thing. I'll go through like each section and kind of either talk through what they look like, or I'll like take pictures on the back of them. Check human anatomy and looking at their eyes.
Okay. Yeah, so actually that exact knowledge is probably going to be very helpful here. We'll probably cover a lot, mostly the same topics. Yeah, and Danny, the tutor, is going to work on some flashcards with you as well. Of course, there's open hours too, so all those things are great for studying. You know, even if you don't have a specific question, if you want to stop by open hours and say, hey, I just want to look over this one section or something like that, that's what it's for. So definitely encourage you to do that. Cool, thanks, Owen. All right, so any other questions? All right. Thanks to everyone who completed the intro survey before class today. I got 15 of them in. I can't match up who submitted it in faces that are here right now, but I think most of you have it in. If you haven't gotten that in yet, do so ASAP and shoot me an email. Let me know that you got it in, because I've already gone through the grades. And so if you submit it now, I might not go back and see. Cool. All right, well, let's test your knowledge of the syllabus. I'm going to pull up this Kahoot screen here. See if this works. I'm going to put the sound. I forgot to set the music to like the good one. It's just going to be the standard music. Here we go. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 of you here today. Go ahead and log in. You can go to kahoot.it and enter that number, or you can just scan that QR code. Is the QR code, is it too small for you to scan from where you're sitting? Or does it work? It works? The QR code work for you? Okay, cool. Oh yeah, I love the random name generator, by the way. So I just put that on for you. Charming gecko, eager, clever otter. Oh, witty pony. That's a nice one. They also totally all sound like clever indie rock band names. Like Gentle Raccoon. Some kind of weird math rock band. Couple more. Let's see. We've got 12, 13. One more person. If you're having technical difficulties, let me know. 1, 2, 3. All right. Cool. Everyone logged in? Decisive dove. I've never seen that one before. All right. So the idea of this is just a fun way to review information on the syllabus. So I'll put up a question. You'll answer it based on your astute knowledge of the syllabus that you read before class today. And then we'll kind of talk about it a little bit, and I can answer any questions or give details. Are you ready? Yeah, let's do it. One more person. Oh, look at that. All right. Someone said totally freak out, but that's all right. Yeah. Basically, if you miss a class in person, as I said, we're all going to have days we miss throughout the semester. Watch the recording, talk to classmates to borrow some notes, talk to me, go to open hours, and you'll be able to make it up. You can also make up in-class activities. You'll see that we'll do a lot of activities in class with like a QR code and a little survey. You can make those up afterwards. You just need to communicate with me, because if I've already graded them, then I might not see your submission. But if you miss a class and get it in within, reasonable amount of time before the next class, then that's just fine. Sound good? I don't know who picked totally freak out, but yeah, don't do that. It's not worth it. There's bigger things to freak out about than missing human anatomy. So look, early on the leaderboard is Helpful Emu on top. All right, let's see who can take down Helpful Emu. Yes. All right. Everyone got this. As I mentioned, the textbook is really important. I'm trying to make it as accessible to everyone as I can. There'll be a lot of readings in it. I guess technically, if you steal it, you don't need to buy one, but I'm not recommending or endorsing that. There is a copy available in Beagley Library on hold. Also, remember, if you want to get, I have the 9th edition. It's the official textbook, but an earlier edition, like the 8th edition, will work just fine. A few of you have like I think it's called the Global Edition or something like that. That's fine too. Just note that if you get an older edition or a different edition, the exact page numbers might differ a little bit, but as long as you read the associated section, then it'll be fine. And I'll always give your reading assignments by like section, read section 3.2 covering the nerves of the eyeball. And then I'll tell you what page numbers. So you can always get that. So let me know if you have any questions, but I want to make sure that the textbook is easy for everyone to get because it is super important. It will help you a lot. Sound good? All right, let's see. Was it Helpful Emu? Oh, still at the top. Look at that. And Witty Pony's catching up. Who's Helpful Emu? Nice job. Witty Pony, though, is only 2 points behind you. Who's Witty Pony? Nice. All right, let's keep going here. We just talked about this one. All right. Somebody said never this stuff. I'm impressed if that was you and you said never. Yeah, we just talked about my best approach for studying, so keep that in mind. Let's see, Helpful Emu. No, Witty Pony. Oh, man, look at that. Helpful Emu's down to 4. Good. Bring a notebook to class. A lot of you prefer to do notes digitally or whatever. However you want to do it's fine, but you will be taking a lot of notes, so be ready for it. It would not be incorrect, I think. Although, today, I guess it would protect you from the sun. It'd be more like a parasol kind of thing. So, no, fair enough. Pencil sharpener won't be the worst. Telescope's not going to do you a lot of good in here, to be honest. Gentle Raccoon stepping up. Look at that. 1 point difference. Witty Pony. Who's Gentle Raccoon? Nice. All right. All right. Office hours are definitely not for losers. Starting Monday and every Monday after, I'll be in our lab room. So that's Science Center 301 from noon to 2. And the idea there is just open hours, drop in. Again, you don't need an appointment. And I have them in the lab room because I have models and specimens and things like that. So it's easier to review course material. And then also if a lot of you come to it, there's more space and everything than my office. So every Monday, noon to 2. Of course, if you want to meet outside of those hours, just let me know. We can make that happen. Sound good? All right. Nobody picked Saturday, and nobody said office hours were for losers. That's good. Witty Pony back on top. All right. We got Clever Otter is shooting up here as well. Who's Clever Otter? Nice, Clever Otter. Good. Yep, visible body. I had on the assignment sheet for today, your class program for today to sign up for it. I just want to acknowledge that it is an additional cost for this class and recognize that. I've kind of debated about how to approach that. The good thing is that you get it for 2 semesters, which actually maybe for this class isn't particularly useful. For students in like human anatomy and physiology that then go on to take human anatomy, so you can use it for both classes. But we will use the software a lot, and students have given me feedback that it is really useful and helps them learn a lot. And so for that reason, we'll continue to use the visible body software. It has really nice interactive kind of 3D model interfaces. It has nice quizzes to practice and some other resources that students have found valuable. So we'll continue to use it. If you haven't registered for Visible Body yet, I didn't log on this morning to see how many of you are in the class, but I have that included for your class program for Monday as well. So do it before Monday so you can get ready for our first lab, which is on Tuesday. Any questions? Oh, look at Helpful Emu has like the fire. That's impressive. Silver Cat is catching up though, coming in the leaderboard here. Who's Silver Cat? Nice. Good. Yep. There's 3 lecture exams throughout the semester. If you look on your syllabus, you have the approximate dates. I have it like within the week that we'll have it. Note that they'll take some will be on Blackboard, some will be in person, and I will announce that well in advance. So you have 3 lecture exams throughout the semester. Not 4. I used to do 4, but it's too many. We don't need 4 exams. Groovy Pelican on top. I don't think who's Groovy Pelican. Nice. That's a good moniker as well. It's like your hip hop name. All right, what is not true of lecture quizzes? So it is true that quizzes will take a variety of formats. It is true they're worth just under 20% of your grade. And it is true that you'll know about them ahead of time. It's not true that they'll be a surprise. I'll always announce it generally a week or so in advance and indicate what the format for that quiz is as well. I will mix it up, but you'll know ahead of time what that will look like. So, Lovely Frog's got the little flag. Who's Lovely Frog? Nice. Wow, that was quick and easy. Good. Don't need to talk about that. Look at that. Nobody changed their position there from that question. That's interesting. All right, let's keep going here. Good. Anytime you have a question, just raise your hand and we'll stop. Your classmates may or may not think you're a nerd, but it's only so much you can do about that in life anyway, I've found. Good. Look, everyone's exactly the same again. That's uncanny. Good. Let me know anytime if you decide you want to go by a different nickname or something like that. I often see like professors like say in their syllabus, like, tell me in the first 2 weeks. And obviously it's important to know early to address someone by their proper name or pronouns. But at the same time, it's not like after the first 2 weeks of class, I'm incapable of changing something or something. So anytime you prefer me to refer to you differently, just give me a shout. And I have that information from your intro survey as well, just your preferred nickname usually. So I get all the names on the like your official legal name on the course roster, but that's not often what folks like to be referred to. Cool. Any questions so far? I'll pause for a quick second. We're good? All right. We're looking good here. That's really weird that it's just not changing at all. Okay. Yep. So there's 2 lab sections, one on Tuesday morning, one on Tuesday afternoon. If you have a conflict with your lab section, let me know in advance, and it's generally perfectly fine to attend the other lab, especially with this class. This is a really small class. It's actually about half the size of the class that I taught last semester, which means labs are really small, so we've got a lot of extra space and flexibility. So both labs on both days will be the exact same thing. If something comes up and you just need to go to the other one, just let me know and we can make that happen. Again, not who's Charming Gecko? I don't think. Yeah, nice. You're stuck in second place though. Oh, this you should. I'll get this one right. Good, good, good. All right, everyone got that one. Good job, class. That's like a pre-test. I'll ask you this question again at the end of the semester and see if you've learned anything throughout the semester. But it doesn't look like you have much to learn. Groovy Pelicans on top and with the fire. You don't see that very often. That's impressive. No, don't show up to my house. One of you gonna be like Tuesday morning, you're gonna come over. I'm ready for that. Yep, we're in Science Center 301. So it's a really big, nice lab room. It's in the other building of the Science Center on the second floor. You'll see it next week. So. Sound good? Oh, wait, do we have one person? Yep, 319. 319 is actually the lab where anatomy and physiology is. It's just down the hall. So this is close to correct, but it's 301. Definitely not my house. Champion Bat rising up, third place. Who's Champion Bat? That's a new one too. Nice job, Champion Bat. I feel like you kind of have to win with a name like that. Like it's built into your name. Good. Oh, that makes me sad that two of you chose that one. I do care what you have to say. And of course, you can tell me in person, but if you have something you'd rather share anonymously, there's a link to an anonymous survey both on the class Blackboard site and from the syllabus. So do that. If you can find a payphone to disguise your voice, you're welcome to that. But oh, I hope you weren't serious when you picked that one, or you picked it on accident. It'd be terrible. Cool. All right, we got lovely frog. We got some. Being a herpetologist, I like seeing the gecko and the frog up there. I don't know. We got to take down this pelican, otherwise Dr. Reichert will get arrogant. This information might actually be in the syllabus, but I want to share. I want to make sure you have this info. So. Good, everyone got that right. You're welcome to take up knitting, of course. But the lab room 301 is open. And actually, there's no other classes in there this semester. So it will generally be like accessible for you to go in and review material. It's locked before 8 a.m. and after 6 p.m. If you want to get in there later at night, doubtful many of you want to get in there earlier in the morning. But I can talk to me and I'll give you an access code and you can get in there. We just keep it locked at night in case, I don't know, nefarious people are wandering around the Science Center and looking for human bones. So let me know. But I want to make sure you have access to that room as much as you need. All right, we've only got a couple more questions here. You're doing great. Glad to see the gecko and the frog. I'm rooting for you. No offense to the pelican, which technically is a reptile. So, you know. Good. His name is Danny. Wow, Lizzo that'd be great. I mean, I'm sure Lizzo, LeBron James, or Hulk would all do a great job, but Danny is your class tutor. He's going to get some information to the class on tutoring sessions and details soon. I'm going to use the information from the intro survey and figure out kind of times and formats that work best. Sound good? Cool. Charming Gecko has the highest answer streak of 17. Nice job. This is getting closer now. Look at there are not many points separating 1 and 2 there. Woody Pony's back on the leaderboard too. Good. So while I endorse you doing all these other things are great, We do not have extra credit in this class. So I just want to make this explicit from the beginning. All of the course points are assigned and laid out in the syllabus in great detail. I won't go over them here. But for a variety of reasons, I don't think extra credit's a good idea. And actually have a link to a little essay from the syllabus about reasons why it's not really a good or equitable idea. If you have questions about that, happy to talk about it more. But generally, I think it's a better policy to have everything laid out ahead of time. You know exactly how you'll be scored on all of your assessments and exactly how your final grade will be tallied. Nobody is going to bring snakes and bring them to class. That's too bad. That would be, if any of those things would get you extra credit, it would be that one. All right, a couple more questions. Good. Avoid yelling, please. Definitely the best email. As I said, I'll get back to you pretty quickly on email. We still have the leaderboard here. Silvercat's got the flame. Wait, I don't think I asked who is Silvercat. Did I ask that one earlier? Oh, that's right. Yeah. Okay, gotcha. Good. We will do a lot of class activities. They're going to be goofy sometimes. We'll get up and do stuff around the room. I'll ask you to like act things out or make a skit or something like that. Often it's really kind of weird and embarrassing, but it will help you remember the course material. So we'll do that kind of sprinkled throughout lecture at any given time. All right, let's see what we got here. Groovy Pelican still holding on to the lead. Couple more questions. All right, so the exact percentage point isn't terribly important. What's important to note is that just under 10% of your grade is participating in all of these in-class activities. Now, I want to emphasize this, that 10% of your grade, or let's be precise, 8% of your grade doesn't seem like a whole lot. But if you think about it, that's the difference between, let's say, a 94%, which is an A, and an 86%, which is a B, right? So that's a pretty big jump. It's almost a whole letter grade of difference just from being in class and participating. So these are all activities that, like, if you do it, you get full credit, usually 2 or 3 points, class points. And if you're not there, you don't make it up afterwards or do it, then you don't get any credit. So just keep that in mind, that a pretty decent amount of your class grade is just being here and doing the things with us. Let's see, and some of you chose 12.5. That's pretty close. That's reasonable if you don't know the exact number. I think we are on our last question here. Helpful Emu just stepped up to the leaderboard. We've got 2 birds and a lizard and then a frog, mammals, whatever. All right, last question. If you choose the wrong answer, you can't go back and change it, can you? You're committed to that. Yeah. Good. So we have 2 lab practicals. The first one is on the syllabus. It's in October. I don't remember the exact date off the top of my head, about halfway through the semester. The final one is during our scheduled final exam period, which is on the Tuesday after classes end. I really like to use that big chunk of time for the lab practical. And so it works out better logistically that way. And I'm making sure you all know this now, because please make sure that date and time is on your calendar for this class and all of your classes. Make sure you know when that final exam period is so that you can make arrangements, for example, not to leave campus or anything like that before that exam period. And we'll have our lab practical then, not the lecture final. So I think that was the last one. Let's see who the podium is going to be. Groovy Pelican. Who's Groovy Pelican again? That's Zoe, right? And then Helpful Emu, that was Veronica. Nice job. And then Charming Gecko stepped up. Is that you, Morgan? Yeah, nice job. I like the reptiles took the chance. All right, I'm going to leave this up here for Dr. Reichert so he can see the lizard beating out the 2 birds. And I'll just go like that and make him feel bad about his career choice. Okay, great. Any questions about this or anything else on the syllabus? Please. Great question. Is visible body only used for labs, or will we use it in lecture? We will only use it for labs. Now, having said that, there's some overlap in content between what we cover in lecture and what you study in lab, so it will still be helpful for the lecture material, but all of the assignments and the quizzes will be assigned through lab for visible body. Yeah, thanks for asking them. Other questions? Get back to Roy Gilmore. All right, well, let's dive into some course content. We got a little bit of time. We probably won't get through all of embryology today, but we'll get through what we can and wrap it up on Monday. So first, to define this, we see a nice scanning electron microscope image of an 8-cell human embryo, obviously very, very, very early in development. And this is an example of what we will study in embryology, which is the study of the origin and early development of a single individual. So generally, the time between conception and birth for humans, or in the case of like egg-laying organisms, it's usually the time between fertilization and hatching, something like that. And so why do we care about embryology? There are a number of reasons. First is that studying embryology actually helps us understand the evolutionary past. So, for example, all of you at one point in your very, very early life had a tail. That's kind of neat because a lot of organisms that are closely related to us, in fact, most of the other mammals, except for the great apes, have tails as well. Now, your tail disappeared rather early in development, but understanding how there's similarities in the way organisms develop gives us information about how those organisms are related in an evolutionary sense. There's also lots of embryo-related questions for those of you that are taking some standardized tests like the MCAT, the GRE, et cetera. If you're looking to go to med school or something like that, there's questions about this on there. That's not really a good reason to care about something to do well on a test, but in a practical sense, it kind of is, because if you're looking to do that as a career, you want to score well on those exams. And so I want to share some of that knowledge with you in here. Furthermore, the last point here is that exposure to something called teratogens during development can lead to a variety of anomalies in adults and children. So for example, fetal alcohol syndrome is the most common form of this in the US. And so by understanding how our bodies develop early, we can understand how that might affect the body later in life. Now, I think it's important here to put a disclaimer here. I really don't like the term teratogen. I'm going to teach you this term because you might come up, like, for example, in an MCAT or something like that. It's the medical term that's commonly used. But I personally don't. I think we need a better term because it's derived from the Greek word for monster and basically means like monster creating. Like the suffix gen here means to create something, as in like the genesis of something. And I really don't like that because I think it's a huge disservice to folks that have suffered from the effects of teratogens early in development. And so I want you to know that term, but I also want to put a disclaimer on it that I think it sucks, and I don't think that we should refer to people as that. So let's talk about the 2 stages of prenatal development. So we're going to break up this period into 2 time periods. The first is called the embryonic period, which is the first 8 weeks. So from fertilization, when the sperm meets the egg, until 8 weeks later. And then we have the fetal period, which is the remaining 30 weeks of development. So humans, gestation period is about 38 weeks, give or take a bit. There's lots of variation in there. And we're going to divide it up into those 2 early periods. And you can see on this timeline here, we have fertilization. We have a 3-week embryo that's about 3 millimeters big, 5-week embryo, an 8-week embryo, kind of looks like this. And then after that, we refer to the fetal period after about 8 weeks. Now, the embryonic period is the first 8 weeks, as I mentioned. And this is really important period because this is when your body plan emerges. By 8 weeks post-fertilization, your general body plan is the same that it is right now. Of course, those structures develop and grow and change over time after that, but the basic plan is all there in place. Importantly, it is this embryonic period as well when these teratogens, so things like environmental pollutants, something like alcohol, viruses sometimes as well, can cause damage and change the way that original body plan is formed. That first 8 weeks of pregnancy is also the time of peak nausea and vomiting, so-called morning sickness as well. Actually, a good friend of mine just had a pretty terrible first trimester. She was nauseous for about 10 weeks, she said, from this. And we're not actually sure why this happens early in pregnancy, but when it does happen, it's generally its peak at this early embryonic period. So this is when everything starts to form and get started. All of those structures emerge from these original single cell. Just going to pause there, see what questions you have. Yeah, go ahead, you. Yeah. Yep. And so remember, there's lots of variation, of course, but generally at 8 weeks, an embryo is about 22 millimeters, which is just under an inch. And that is head to rump, basically. So head to rump. So an inch is about 25 millimeters. So it's just shy of an inch to convert. So you'll notice in your textbook readings for this chapter, I skipped a bunch of sections that provide a lot more detail than I'm going to cover in class or I expect you to know. You're welcome, of course, to check those out, but I'm just going to focus on some of the major ideas. And specifically, I'm going to skip over all of this early development, the early cell division, and how the embryo is attached to the uterine wall and all of that good stuff. And I'm instead just going to kind of skip ahead to the major body structures that develop during this embryonic stage. So this is what an embryo looks like at about 5 weeks. And you can see here 3 different colors. And these colors are the same as your textbook. This image is actually from your textbook. And they show the 3 major layers that develop during this time. And I want to talk about each of those in a little more detail. Actually, I'll talk about the names of the layers. Do I talk about the names of the layers in another slide? Yeah, I'll get to the names of the layers in just a second. But what I want you to see here is what's in yellow is called the inner tube. And this is generally going to be your respiratory and digestive systems. Then we have what's called this red section, which is called here a segmented outer tube, but this generally becomes a lot of organs in your muscles, your skeletal muscles especially. And then we have this dorsal hollow nerve tube, which becomes your brain and your spinal cord. And notice here, we also have something in purple that's called a notochord. So in an evolutionary sense, this notochord is what makes us vertebrates is the presence of this notochord. Now, it's important to note that this is not your spine, but this is the structure that appears early in development and actually later becomes the cartilage discs between your vertebrae. We'll talk about this in more detail, but if you hear someone that says they have a slipped disc, that's this cartilage kind of moving out of alignment or becoming inflamed. And those cartilaginous discs are derived from this original notochord. So the takeaway message here is that we have structures that are yellow and red and blue here. And I'm going to show you those same structures in another animal and then in a human. And the idea that what I want you to take away from this is that they're all the same. Those structures develop from an embryo to an adult, but we also see them in other animals. So here's a fish, for example. I'm not going to ask you any questions about fish. I just want to show you this so you kind of have a basis for comparison. But notice that this yellow structure develops into their gill slit, so they're essentially their respiratory system and their digestive tract. This red structure is their muscle, and then we have this brain and spinal cord, the same, and we have this purple notochord. So the same basic structures. They look, they have different shapes and different forms. They're in fish form instead of human form, but those same things that are in the fish are in the human embryo. Also important to note is that us humans and all other, with some limited exceptions during development, all other vertebrates have what's called bilateral symmetry. That simply means that if you look at our bodies, the left and right sides are the same. Other organisms don't have this. So for example, if you take a jellyfish, they don't have I guess they do, they have radial symmetry, or like a sea star or something like that. Their left and their right don't look the same. Instead, they're kind of have radial symmetry, so you can kind of rotate them, or other echinoderms or things like that. But we have bilateral symmetry from early development. So as these structures develop early, they develop generally the same on both left and right sides. Now we bring this back to an adult, and you can see these same structures in the same colors here. Those red structures develop into the musculature of your rib cage, your thorax there, or your thoracic cavity there. We have the digestive tube here in yellow, and then we have that spinal cord in green. So those structures are the same. Now I want to take a step back and show you where those structures come from. So early in development, we have this layers of cells. Again, I'm going to skip over the early parts about how those early layers form, and instead take you right to here, where we have 3 layers of cells that form that are basically on top of each other. We have what's called the ectoderm, the mesoderm, and the endoderm. So those are 3 terms. I should highlight this. Make sure you know these 3 terms. We're going to come back to that. I'll talk about each of them in more detail. But what happens is this kind of cool thing where the 3 layers are just flat like this, and then they kind of fold into each other to create a tube early in development. And that yellow layer you see is now, instead of just being on the bottom of like a cake-like thing, is now on the inside. Remember that yellow layer, the endoderm, forms those structures basically in the middle of your body, your respiratory system and your digestive system, your gut. Then that blue layer is on the outside. That blue layer, the ectoderm, develops into your skin and integumentary system on the outside of your body. Got something that looks like that. And if we look at this at a 24, let me see, do I want to talk about this? Yeah, I'm going to show you this diagram. So those structures kind of fold over each other. And if we cut this along this sagittal plane, so cutting it from head to tail, you can see how those different structures start to form what will become a human body. So you have the head here, and then all of this stuff in blue is what becomes your skin. And also from the ectoderm, we get the brain and spinal cord. So you can see that here. The red layer, the mesoderm, becomes a number of organs, kidneys, for example, and also your musculature. And then this yellow tube in the middle becomes your digestive system. So we have this tube within a tube structure. So what's important to note, we have a tube that goes down the middle of our body, our digestive system, and then we have the rest of our organs forming around that. So technically, things that are in your digestive system are actually outside of your body because they're not within, they're in that tube that's within the tube. They're not in the actual body structures. Of course, we don't really think about that way and in a functional sense. If you swallow something, you kind of think of it as inside your body. But it's important to note that tube goes all the way through your body. And so the rest of your body is structured around that internal tube. Ectoderm, mesoderm, endoderm. You'll see this come up in this throughout the semester. You'll see things with the prefix ecto, meso, and endo. Ecto means on the outside. Meso means in the middle. Endo means on the inside. You'll see those come up again and again. Then from those 3 layers, we have a number of structures that develop. Now, I'm not going to get into, I'm not going to ask you to know a ton of detail about what this looks like. Let me just take a look here and see what time is it. How much do I want to cover? Yeah, I'll keep talking for a few more minutes here. This is a diagram from the book, or a chart rather from the book. I'm not going to ask you a ton of all of the detail here, but here is what I do want you to know. And I'll see, let me write this on the board right over here. That way it'll show up in the video. So I want you to know that from the ectoderm, we get skin and associated structures. So nails, hair, sweat glands, stuff like that. And your nervous system, interestingly, is also derived from the ectoderm. And so that's your brain, spinal cord, etc. And we're going to get into the nervous system later this semester. I also want you to know that from the mesoderm, actually, I'm going to skip the mesoderm for a moment. I'm going to do the endoderm. The endoderm forms the basis of your digestive and respiratory systems. So you'll see this diagram I'm showing you gets into a lot more detail, but this is the main takeaways that I want you to know. Again, endoderm is the yellow layer, right? And so that's the layer that forms the middle of that tube. So it makes sense that these structures that are down the middle of our body would be derived from that tissue layer. The mesoderm is a little more complicated, and you can see that by the fact that we've got like a little list over here for the ectoderm, a little list over here for the endoderm, and then everything else is from the endoderm or the mesoderm. And so you can think of the endoderm as basically, excuse me, misspoke. You can think of the mesoderm as basically everything else. And I want to talk a little bit about some important structures that are included in that everything else. Endoderm, oh, darn, keep doing it. Mesoderm is everything else, which includes your muscles, your skeletal muscles. What else do we have here? Important organs like your gonads and your kidneys. Oops, I can spell. Also includes structures like your heart, which is here, and blood vessels. And then also, I want to note that your skin is actually made up of two layers, an outer layer called an epidermis that's derived from the ectoderm. But there's another layer underneath called the dermis, and the dermis is actually derived from the mesoderm. We're going to talk about that in detail starting on Monday when we get to the integumentary system. I just want to throw that out there now that the skin is actually the outer layer is derived from this ectoderm. The inner layer, the dermis, is derived from the mesoderm. So those are the three layers. I'm not going to ask you, and we're not going to get into the somatic and splanchnic mesoderm. I don't need you to know all of that detail. This diagram is pretty handy, but I do want you to know these major organ structures and which germ layers they're derived from. And we can look at a cross section of a human body, actually of an embryo, which is a human body. If we look at a transverse section, so this is cutting the embryo essentially kind of right here and looking down on it. And you can see, again, these three layers, the blue ectoderm that's forming this outer layer that develops into the skin, and then this inner layer that develops into the nervous system. We have, in red here, this mesoderm that develops into the heart, the kidneys, the muscles, etc. And then we have this yellow tube in the middle that becomes the digestive tract. Go ahead, Abby.
So the ectoderm also forms the nervous system.
Ectoderm is outer layer of skin, nervous system. Exactly. Yep. And that happens. There's a weird fold that happens when some of these cells migrate. I'm glossing over some of those details, but the way that those cells migrate and kind of come inside, so the ectoderm is not just. And we can see this exact same structure carry through in an adult. This is a transverse section of an adult. And you see almost the exact same pattern here. Now, the relative size of things have changed. The spinal cord is relatively smaller. More space is taken up by the mesoderm. The endoderm layer, the digestive tract, gets relatively smaller. But you can see that that structure looks very similar to this in terms of the layers and their placement relative to each other. Now, if we look at a limb, we see a similar structure as well, except there's no endoderm. Limbs are formed entirely from the ectoderm and the mesoderm. So you see the same thing. You see the skin on the outside derived from the ectoderm, and then you see the layers of skin, the layers of bone and muscle derived from the mesoderm. Let's see here. Yeah, I'm going to stop here. We're right at 8:50 actually. We'll pick this up on Monday. I'm just going to end things real quick with an announcement. Remember to check your class program. There's some textbook readings for Monday. There's your Friday Venn diagram. And have a great weekend, everyone. Thanks so much. We'll see you Monday morning.